The session. And we encourage you to post your questions in the Q&A or raise your hand and we can ask you to unmute. The Q&A is preserved and the chat is not. And we have a great selection of speakers talking about transcription and expression. So I'll turn it over to our first speaker, Chen.
Thank you. Can everybody see my screen?
Yes.
Great. Hi, I'm Yu, a graduate student at Cornell University, co-advised by Dr. Andy Clark and Dr. Haiwan Yu. I'm very happy to be here and to share some interesting findings about directionality of transcriptional regulatory elements. Promoters and enhancers are two classes of TREs that serve as major control hubs for gene regulation. Accurately mapping and characterizing these elements is very critical for understanding how transcriptomes are regulated in normal and disease states. Here are some classical features people use to identify active TREs. Both promoters and enhancers are located in open chromatin regions, which contain multiple binding sites for transcription factors. And they are marked by histone modifications, such as H3K4 monomethylation and H3K27 acetylation. More recently, our group and others have observed that, in addition to these epigenomic features, widespread transcription is found in both promoters and enhancers, which can be best detected by GRO or ProCap assays. And it turns out that these nascent transcription events are critical markers of active TREs. Here we selected candidate elements that are located in open chromatin regions and have decent histone marks. These elements are further divided into two groups, untranscribed and transcribed. As controls, we included proximal elements located within 1 kb region surrounding gene TSS, which are potential promoters, while those outside this range are classified as distal elements, which are potential enhancers. Then we used an episomal assay, E*-seq, to quantify enhancer activity of these candidate elements. We can see that untranscribed elements had very little enhancer activity, while the transcribed distal elements displayed a much higher activity rate. These findings demonstrate that transcription is indeed a critical marker of active enhancers. If we further look into these transcription events, we can actually observe different patterns. Here we perform ProCap assay in K562 cell line and included about 20,000 divergent elements for follow-up analysis. For today's talk, I will only focus on the distal group, which are potential enhancers. This is a typical example of divergent elements. We can clearly see two peaks on the opposite strands, which indicates that these RNA polymers too are firing in the opposite directions. This divergent transcription has been well studied and recognized as a critical mark of active enhancers. However, we also noticed that there is another type of transcription where we only see peaks originated from one strand. These induction elements have been overlooked and their identity and functions remain largely unknown. To better understand this category and also investigate the contributors of directionality, we performed systematic comparison between divergent and induction elements from different aspects. First, let's look into architectures of these elements. The meta plot on the right exhibits the average signals across all divergent elements. For better visualization, we have flipped the side with small read counts to the right. In the heat map, the elements are anchored on the midpoint between two prominent TSS, and they are sorted by the distance to the center. As expected, they are centered on open chromatin regions, with histone marks found on both sides. Then we looked into uniduction elements. Since we don't detect any peaks on the other side, here we anchored elements on the center of neighboring DNase peak as an approximation. Compared to the divergent group, induction elements have much lower signals in all these assays. And histone marks are more predominantly enriched on the same side where we detect ProCap reads. Despite differences in the number of transcription units, we found that these two groups actually have very similar sequence content when we zoom into individual transcription units. Here, nucleotides C and G are labeled as black, and nucleotides A and T are labeled as white. For divergent elements, we can see there are two parallel arcs on both sides. While in the uniaxial group, we can see a similar set of arcs present on the side, where we detected procap peaks. To figure out the identity of these arcs, we examined the streamer frequency within 100 BP region surrounding TSS. First, let's look at the prominent TSS on the maximum side of divergent elements, the side with more rate counts. We found that the inner arc represents Tata box-like sequences, while the outer arc represents initiator-like sequences. We observed a similar pattern for minimum TSS of divergent elements and the unidirectional TSS. Given the similarity of sequence content in individual transcription units, we are wondering if there is an evolving process from unidirectional to divergent elements. If this is the case, the sequence gained transcription on the other side during the evolution. We should expect to see the age differences between these two peaks. To estimate the sequence age, we employ the syntactic block aging strategy, which estimates the time to most recent common ancestor using the phylogenetic tree. Here we can see that both divergent and unidirectional groups are older than the random genomic controls, indicating that they may have been maintained for their functions. Unidirectional elements are younger compared to their divergent counterparts. Since we have flipped the side with more rate counts to the right, so you can also see that the side with higher transcription level tend to be older than the other side. Next, we look into two evolutionary metrics. P used to measure interspecies conservation. Here we can see both groups have a highest score in the middle, which contain binding sites for transcription factors. Another metric, CDTS, evaluates more recent or ongoing selection in humans. Lower score means this region is less tolerant to variation. Here we can see a similar pattern as sequence age. Divergent elements are less tolerant to variation compared to unidirectional elements, and the side with more rates are less tolerant compared to the other side. Taken together, there seems to be an evolving process underlying directionality changes. Considering their recent evolutionary origins, we are curious if unidirectional elements have acquired gene regulatory functions. First, we found that unidirectional elements have similar genomic distributions as their divergent counterparts, indicating that they may also play a role in gene regulation. To further evaluate the regulatory potential of these unidirectional elements, we predicted target genes using ABC model based on element activity and 3D content frequency. We can see that there is a higher proportion of unidirectional elements that don't have predicted target genes, indicating that some of them may still be in a less mature stage, though others may have gained regulatory functions. In addition to in silico predictions, we also examined regulatory effects of unidirectional elements using publicly available CRISPR dataset. For instance, the deletion of this element significantly reduced the expression levels of these three target genes by 30 to 40%. In addition to the potential evolutionary process underlying directionality changes, we are wondering if there are other contributors of directionality. We compared the protein binding profiles of these two groups using ChIP-seq data. We can see that divergent elements show greater enrichment for most proteins surveyed, while CTCF and cohesion components are more enriched in unidirectional group. So we are curious if CTCF blocks transcription initiation on the other side. To address this, we performed the ProCap assay in a CT16 cell line before and after CTCF degradation, and we do observe hundreds of elements with significant upregulation after CTCF loss. For example, here is a unidirectional element bound by CTCF that shows increased transcription on the other side after CTCF degradation. And we noticed that this CTCF motif is located downstream of the blocked TSS. Then we examine the transcription changes relative to CTCF motif in all elements that show upregulation either in the minimum or maximum site. This rule holds true that CTCF serves as the blocker when located downstream of TSS. Here, downstream refers to the direction where RNA polymers 2 is moving toward. Interestingly, we found that CTCF can also activate transcription in some cases. Here is one example that shows downregulation after CTCF degradation. And you can see CTCF motif is located in the upstream region this time. We observe the same pattern when we examine the CTCF positions in all elements with downregulated transcription. Taken together, CTCF exhibit dual function on transcription initiation in a position-dependent manner. It serves as the blocker when located downstream of TSS and as an activator when located upstream of TSS. To further test whether this dual function impacts directionality, we use the metric directionality index to evaluate whether an element is more balanced with similar rates on the forward and reverse strands or more skewed toward one strand. As shown in the previous example, before degradation, transcription is skewed toward the forward strand. After degradation, it becomes more balanced due to increased transcription on the reverse strand. This trend is consistent when examining changes of directionality index across other elements. To systematically test if there are other factors with similar dual function, we trained a ProCapNet model using ProCap data of wild-type HCT116. ProCapNet is a deep learning model that can predict total counts and transcription profiles at a base pair resolution for a given sequence. We use the model to predict the rate counts based on the reference and the mutant sequences with the motif of interest deleted. Consistent with our experimental results, we see that CTCF exhibits a dual function in transcription. And then we can see a few more motifs showing this feature. Let's take a look at this AP1 motif, for example. The first track shows the observed ProCap signals, and the second track shows the predicted signals, which closely resemble the real data. We can also see that this AP1 motif is located downstream of the TSS on the reverse strand. After in silico deletion of AP1 motif, we can see increased transcription on the reverse strand. Here is another example where AP1 is located in the upstream region. After deleting this motif, the transcription level is significantly reduced. In summary, induction elements are a true biological entity with distinct features. There seems to be an evolving process underlying directionality changes. Moreover, we found that some TF exhibit dual function on transcription initiation and directionality at TREs in a position-dependent manner. Finally, I'd like to thank everyone who contributed to this project. Thank you all for your attention. I'm happy to take any questions.
All right, we have some time for questions. Are there any questions? Either you can type them in the Q&A or raise your hand. I have a question.
Okay, good.
How does the syntenic block age estimate work? Can you tell us a little bit more about that? How did you estimate that these were younger?
Yeah, let's go back to the slide. Yeah, we mainly use this strategy described in this paper. So basically, they download the syntenic blocks from the UCSC database. Then you can overlap your element with these blocks, and then you can check the most recent common ancestor for that sequence. And so here basically further categorized this species into 10 age categories, and each categories correspond to a specific number. like millions of years ago, then we can quantify this, quantify the age of all these elements and compare these two groups.
Great. Thanks so much. I think we should move on to the next talk. So thanks again for a really interesting talk.
Thank you.
All right. Do you want to share your slides, Katarina?
Yes. Okay. I just wanted to make sure it was me. I thought it was.
Yep. Yep.
You guys see that okay?
Yes, we can. So
Okay, awesome.
yeah, take it.
All right. My name is Katarina Mapes. I'm a PhD student at Oklahoma State University. I work in the Department of Integrative Biology with Dr. Gwyn Wogan. I don't have as much amazing data, but I do have some research to share with you guys today. So my topic is the aging game. See if it'll let me. All right, there we go. So what I'm going to be talking about today is aging. So as far as aging is concerned, how do you die of old age? I feel like that's not something people think about a lot. You think about all these other ways that you could die, but you don't often think about the way we all want to die, which is just of old age, right? So what actually happens? What are the biological mechanisms behind this? So we have our chromosomes, and on the end of each of the chromosomes, we have something called telomeres. And these are repeat tandem sequences that are slowly degraded every single time our cell replicates. This is the pattern that we see in most mammals. So every single time those cells replicate, they're slowly getting degraded. Now, there are some other enzymes and things that are able to come in and help repair some of that degradation, but they're not able to fully keep up with the rate of degradation. But there are actually some species where we're finding different patterns and they're displaying extraordinarily long lifespans. So as you can see over here on this chart, we would expect a regular mammal who's following these patterns, like humans per se, for their telomeres to degrade over time, obviously, while their chromosomal length is remaining consistent. But in some species, we're finding that they're maintaining their telomere lengths over time, which is indicative of healthy aging in adult stem cell compartments. So what species am I talking about specifically? My study species, Ictobius cyprinellus, commonly known as the big mouth buffalo fish. It's distributed throughout North America and is native to where I am here in Oklahoma. We previously believed they could live to be about 25 or 26 years old, but Lackman at the University of Minnesota found that their populations were living up to 125 years old, which was really extraordinary. Like I said, these are spread throughout North America, but we do have a pretty predominant population here in Oklahoma. So that's part of what I'm going to be looking at. Not only are they really awesome because they live so much longer than we thought that they would, they're also really commonly targets of bow fishing. So some people aren't good enough with just a pole, so they had to like shoot a bow and arrow in the water. All jokes aside, I know that some people, it's a sport, but oftentimes they are targeting the larger individuals. The larger individuals of buffalo fish in this case are the pregnant females. Females and all buffalo fish have delayed sexual maturity, and they also are very selective of when they produce. So they don't reproduce every year. So when we're killing these pregnant females, it is probably having a significant impact on the population. So just the preliminary part of my work that I'm going to be doing is helping the Oklahoma Department of Wildlife Conservation assess the population demography, learn about their ages, and kind of on their end, they'll take my research and say, like, does there need to be some sort of change in conservation status? Additionally, when they are getting rid of them at these bow fishing competitions, they're not a fish that is eaten. So being around Oklahoma here, it's not something I would ever refer to them as, and I hope by the end of this I can change your mind, but people refer to them commonly as like trash fish. People don't eat them, and a lot of the times, the reason they're not eaten is because people can get sick. So there have been a few studies that have linked them to causing rhabdomyosis, and all over, they're not a very delicious fish to eat. So whenever they're getting rid of them at all of these bow fishing competitions, we were able to partner up with them to take their fish after they were done with them. Once they weigh them and take their pictures, they sadly just throw them all away. So we were able to collect genetic samples from all of these, not only at bow fishing competitions, but lots of opportunistic collection, including when they've had to drain some dams here in Oklahoma, we've been able to collect. We've collected and extracted 1,200 buffalo fish fin clips and about 60 gonad oocyte samples. and otoliths from each of the 1,200 individuals. So otoliths are these stones that are inside the head of the fish and other organisms, and they aid in buoyancy and environmental detection. But similar to if you were to cut open a tree and look at the stump, you can see the concentric rings that correlate to the amount of years that they've been alive. So we're able to get concrete ages based off that, which is going to be provided by the wildlife department. And it just so happens, at first I wasn't sure what I was going to do with these 60 gonad oocyte tissues that I took. I just thought they're getting thrown away anyways, might as well take some. And it turns out that the bigmouth buffalo fish is suspected of having this one specific nematode parasite that's only in their gonads. So I made a parasitologist very happy, telling them, well, I've got a ton waiting in the deep freeze. So we did a lot of opportunistic collection. And then this is just a slight little map of our collection areas distributed throughout Oklahoma. I wish there was better water overlay, but a lot of these are smaller rivers, lakes, and dams. And so my dissertation is going to be addressing the following questions. I'm going to start off with just addressing, are there, does shared evolutionary history drive patterns of longevity among fishes? So how I'm going to be doing this is I'm going to be taking, or have been, an age database as well as fish base and do a cumulative analysis of what I call centenarian fish species, although I've had a few other people be like, I don't know if I would say centenarian applies to fish, but we can argue about that later. And so what I'm going to be doing is taking these databases. It's a very simplified phylogenetic tree only containing species that are 85 and above, or 80 and above at this point. But I'll have a big tree and we're going to delimitate a bunch of different traits and see if there's any phylogenetically conserved traits throughout evolution that have contributed to the centenarian status. And so you can see up here, we've got in our top right, this is our bigmouth buffalo fish. And we've got a really wide variety of things that can live this long. So we've got from the buffalo fish to some really small Oreodories, all the way to Somnonosus microcephalus, which is the Greenland shark, one of the most well-known fish sharks out there because they can live almost 400 years, which is really amazing. They're known to live in really cold waters. So there are other traits we're going to be looking at. Where do they live in the water column? What temperature do they prefer? Mode of feeding, things like this, and see if there's any conserved traits throughout these centenarians. Now, once we're looking at that, which will contribute to our overall knowledge of the buffalo fish, I'm going to be specifically diving into my work on the buffalo fish. And what I'm going to be asking is, does the Oklahoma population of buffalo fish exhibit a similar lifespan with similar telomere maintenance as found in the Minnesota populations? So we have a cytochrome B barcoding that we've been doing on these 1,200 individuals. It's going to give us which species of buffalo fish they are. So there are 3 species, the bigmouth, the smallmouth, and the black buffalo fish. Only the bigmouth is suspected to have shown this pattern of telomere maintenance and the extended longevity. So it's important to delineate between them because just looking at them morphologically, it's extremely difficult to tell the difference. And so we're looking at not only cytochrome B for barcoding, but also confirming it through dd-RADS. Then what we're going to be doing once we confirm who are the bigmouth buffalo is actually doing a comparison of their telomere lengths. So I've created, and should be here in a few days, very excited, a plasmid that we're going to be using to do a telomere single copy ratio. And so it's going to be a qPCR. This is our plasmid standard using beta actin from carp and just a repeat telomere sequence connected to a backbone. And we're going to be able to get telomere lengths for each of these individuals and make comparisons between otolith age and telomere lengths. Now, I felt I needed to take it a little bit further, and I said, okay, great. They're maintaining their telomeres. It would be great if they do that in Oklahoma as well. And if they do, what else, right? How are they doing this? That was part of my question. And so earlier I was talking about our telomeres and how there's this thing called telomerase. And what that does is helps repair some of the degradation from cellular replication. TERT is the telomerase reverse transcriptase gene, which upregulates telomerase. It tells our body, make telomerase and release it, basically. And so I'm curious if that has anything to do, the expression of TERT has anything to do with the ability to have this telomere maintenance. Additionally, I'm going to be looking at FOXO1, which is a group of evolutionarily conserved transcription factors that heavily influence processes throughout the entire body, but specifically aging as well, and seeing how, through an RNA-seq, how the expression of these levels differ between juveniles and adults and throughout the species, throughout the different populations. How does it compare in Oklahoma to Minnesota? And so my main goal here is to investigate what these biological genetic mechanisms are behind their telomere dynamics. Lastly, I just had to take it a little bit further and say, well, does it work for anything? We've seen in some other papers, they've been able to do telomerase injections in zebrafish. And I was curious, I had a few things. I was like, well, you only have male zebrafish. So there's a lot of telomere disorders that can be more likely to affect one than the other. And additionally, they were missing some other genes. And so I'd seen other papers where they had done a knockout of FOXO1 and how that affected the aging. But I said, why are we not combining these things? They seem like two really, really powerful genes as far as our telomeres and aging go. So what I'm going to be doing is some trials with zebrafish, and we are going to be creating knockouts of FOXO1 and TERT in combination and in separately, and comparing them to our wild type group once we do a recombinant mRNA vaccine containing both the FOXO1 and the TERT. And so the goal here is to say, what can we learn from buffalo fish and their telomere dynamics? It's awesome that they maintain their telomeres, they live so long, but is there anything we can learn from them? Is there any way that we can improve life for other people, including those with pulmonary fibrosis or any other telomeropathies, short telomere syndrome? There's a ton of telomere biology disorders that predispose people to having shorter telomeres and a shorter lifespan. And not only people, there are also other species that are experiencing early senescence. I've even had people come up to me and say, have you looked into golden retrievers? Their lifespan is shortening and all of these things. So it's very interesting when you look into that. And it's one of those things you may not think about till it affects you. But the goal here is not to live forever. It never has been. That's not what anybody wants to do. But the goal is to see how can we make those who are predispositioned to have shorter telomeres live longer, live just as long as everybody else. And so that's the goal here, is to see how we can use information learning from the buffalo fish for possible upstream gene therapies for those with telomere disorders. That's all I have for you guys today, but I'm very open to any questions and excited in the future to come back and share some results.
Great. Thank you so much for that. We have plenty of time for questions, if anyone has any. Yeah, go.
Yeah.
I have a quick question. You showed that there are like a lot of fish species with maximum age already measured. So I wonder, just if you calculate the phylogenetic signal, like how much would be the phylogenetic signal of this trait?
Yes, and are you asking, is that what I'm planning on doing? Hello?
Sorry. I mean, say how much? Let's say how much variation in lifespan is explained by like the phylogenetic relationship. So like have you tried calculating the quantitative statistic for this?
Not yet, not yet. So we're in very preliminary things right now. And so we're just kind of calculating getting all of the trees together. I haven't gotten to run any trait data or phylogenetic signal data yet, but that is the goal. And so that is an important thing to think about, how important is the lifespan itself.
Yeah, thank you.
Thank you.
Are there other questions? I had a question about comparing the Minnesota and the Oklahoma fish. Do you have a prediction for what might be different about them?
I don't predict much is going to be very different, but it's more we have to confirm it. So we know that these Minnesota populations are experiencing this, and we do expect the buffalo fish here to show the same pattern, but it's only been shown in the Minnesota population. So I think to really dive into this, the first question is, well, are they doing the same thing? So I'm not expecting a huge difference. The only place where I'm curious to see if I see a difference is that the Minnesota population was heavily skewed towards adults. They were having a really hard time finding any juveniles. So like maybe we don't know everything about their spawning and things like that, or possibly there's some sort of reproduction event happening. But they were having a really hard time. They're like, we can't find any juveniles. So that's something that may be different. Like maybe we'll have a greater population of juveniles in Oklahoma. That's something that could be interesting to look at. Also, they said their maximum age there was around 120, 125. I'd be interested to see if we have anything older or younger. And then I think the last thing is a lot of people will say Ictobius niger, the black buffalo fish, they'll say like it doesn't exist, or like it's a hybrid of the two, or this, and that. And so I think it's interesting because as far as cytochrome B barcoding has gone, I've gotten quite a few black buffalo fish back, but they're so similar genetically. It's like few nucleotides off in the sequences. So that's why we're doing the DD ride, just to confirm.
Okay, great.
Yeah.
I think we'll have to wrap it up because it's time to move on to the next stop. But yeah, I'm excited to see what you find.
Thank you.
All right. Does our next speaker, I think it's, yeah, you want to share your slides? Excellent. All right. I'm looking forward to your talk, so take it away.
Hi everyone. This is Rex. I'm glad to be here introducing our recent work, which is a general evolutionary model for the emergence of evolutionary novelties from serial homologs. Okay, the subject is serial homologs. They refer to repeated body parts of organisms with shared ancestral developmental programs. By developmental programs, we mean gene expression during development. Here's a schematic illustration. So you have some developmental program repeatedly executed during development, resulting in these repeated parts. But then during evolution, this ancestral developmental program may be modified such that the same part will develop into different forms. Serial homologs are important because they are the basis for a lot of evolutionary novelties for morphological characters with novel forms and functions. Here are some examples of serial homologs that develop into different forms. One classic example is body segment of arthropods. Arthropods of different groups consist of segments of different sizes and morphology. And other example are the appendages attached to these segments. Among them is insect wings. While some insects, their forewings and hindwings are more similar to each other, well, they are pretty obvious some kind of repeat, while in some other insects, they have different morphology. And in certain groups, there are wings that evolve into very specialized forms, such as the hot ears of dipterans and elytra of beetles. Vertebrates also consist of various kinds of repeated parts, such as our vertebra, which are regionalized in different ways in different groups. And in some species, they are modified into very specialized morphology. And our limbs, forelimbs and hind limbs, are serially homologous. They have the same basic skeletal structure. They have relatively similar morphology in some species, but while in some others, they can be very different in terms of size and digit number. In addition to these complex morphological characters, cell types are also essentially serial homologs. So cells are essentially repeats as well, and these different cells express different genes that produce different phenotypes. At this point, the studies in developmental biology have unveiled some principles of their development. Importantly, there exists a hierarchical gene regulatory network regulating their developmental processes. So there exists a gene that produces a character state, which is a specific phenotype we see. And there are also genes that specify character identity or what a body part is. And what does that mean? So again, in the example of insect wings, so forewings and hindwings are two distinct identities. So the forewings of these three insects are homologous organs that have the same identity but different states in these different species. And similarly, the hindwings. The nature of identity, character identity, has been demonstrated in some developmental studies. with this case of UBX expression being a prime example. So this gene specifies hindwing identity, and the loss of its expression will cause the hindwing to be replaced by forewings. In Drosophila, then it's the halteres replaced by wing blades, whereas in the beetle, it's the wing blades replaced by elytra. So in conclusion, this gene doesn't specify any specific wing morphology state, but instead hindwing identity. Similarly, tetrapod limbs are also regulated in a similar way. So we know there are genes specifying forelimb and hind limb identities respectively, and there are other genes specifying their states. So what we know now, well, as mentioned, is some principles of development. And we still need to better leverage this knowledge to understand the evolutionary dynamics. Importantly, we still need a modeling framework under which we can perform quantitative investigations for how features of the developmental processes can constrain or bias the course of evolution. So that's what we are doing here. As the initial step towards this question, we propose this generalizable modeling framework that involves two categories of genes regulating the development. So there are these identity determination genes and state determination genes. So the IDGs are regulated through a local transregulatory environment, namely levels of morphogens in a given body part, and they will regulate downstream state determination genes, which are effectors producing the character state we can observe. And this can be represented mathematically using systems of differential equations. So briefly, we have parameters that capture the transregulatory environment, these elements of the identity genes, and also gene product decay. Then for the state gene determination gene expression, so it's determined collectively by the regulators and these interaction parameters. So solving this, you get the equilibrium expression levels. And all genes together can be represented using these matrices. So I'm not going into greater details about this mathematics. And for the rest of the talk, I will focus on some applications of the model. So I examined the two types of evolutionary changes and used the population genetic simulations to demonstrate the dynamics under developmental constraints. So I examined the conditions under which these evolutionary changes are likely to happen. So the first kind is divergence of serial homologs with conserved identities. So these IDG expressions conserved over time, but there will be cis regulatory mutations affecting the SDG expression in specific body parts. For example, like this one, it happens in this gene's regulatory element. So it will only affect the phenotype of a certain character where it is expressed. So you can imagine these as tetrapod limbs. We examine a series of evolutionary scenarios. So first, there are different levels of developmental constraint. So that is, the two identity genes can share some, they may not share targets. In that case, there's no genetic covariation, but maybe they do share targets, in which case there will be a genetic covariance between the two body parts and causing them to tend to co-evolve. We also examined some different scenarios of directional selection, including this case where one body part is on the directional selection while the other is on the stabilizing selection, or both on the directional selection and it's concordant, or it's divergent, selecting for a state where the two body parts have very different states. at last, there are different scenarios of correlational selection characterized by the adaptive zones. There are different directions of correlation. So together, there are 9 combinations of directional and correlational selection. And here's a summary of the simulation outcome. So the x-axis is the number of shared targets by the 2 identity genes, and the y-axis is the phenotypic divergence between serial homologs. The blue line is the expected amount of divergence upon optimization of the phenotype. So there are some scenarios in which they do undergo substantial divergence, and certain parameter zones under which this happens faster. Namely, when the 2 identity genes don't share too many targets. The other kind of evolutionary change is switching of character identity of a given body part. So in this case, there are mutations that will shift the transregulatory environment and turn on and turn off the identity genes. There would also be these mutations affecting the downstream, affecting the phenotype of a given identity. So for the IDG expression, we consider this threshold model. There's the cutoff of the morphogen level, below which one identity expressed, beyond which the other identity expressed, and the ancestral level is set here. And we considered some different scenarios of selection. So the optimal phenotype can be the phenotypic state produced by either identity. It can also be something in between. But because the effect of identity shifting mutation is like this, so whenever the optimums are in between them, so it will overshoot. So what difference is that gonna make? But it turns out in these 2 scenarios, identity shifting is about equally likely. Well, of course, it can happen and favored by selection when it produces the phenotypic state that is optimal, but it doesn't have to be. It can also be something in the vicinity, and that is enough to make it equally likely. So these are some very initial steps, and that's what we have done for now. But in the future, there would be more to study under this framework. So for example, the number of serial homologs of the same type. One example is the number of vertebra. Say mammals all generally have 7 cervical vertebra, which is a strong constraint. Even giraffe have 7 very long neck vertebra, while in sauropsids, it's very variable. They can have many more. So what's the principles underlying the change of number and the variation across lineages? So that's something we want to explore in the future. And another general problem is how does the movement of the optimal phenotype over time affect the evolutionary dynamics? So the adaptive zone can shift, causing different clades to have different adaptive zones and different dynamics of their own. So the effect of moving optimum may be important to explain some very long-term evolutionary patterns. So our preprint is posted introducing this modeling framework. You're all welcome to check it out. Unfortunately, I'm not gonna present this in the in-person meeting for what reasons. So you are really welcome to check out the preprint or reach out with questions or thoughts. Thank you for coming and questions welcomed.
All right, that's a great talk. We might have time for one quick question. Anyone has one? You're also welcome to post. Oh, yeah, I see a raised hand.
Yeah, I have a quick question. So first comment, when you mentioned the ultrabiothorics expression in the Drosophila, so I don't think this Haltier had become another pair of 4 wing. It's the 2 thoraxes over there. But the other thing I was wondering is whether your model it's really generally applicable for different type of serial homologs because they may be underlying, I mean, there may be different genetic mechanisms underlying the formation of different type of serial homologs. What do you think of that?
Yeah, that's an important point. I can't say for sure how general it will be, but I think this framework does capture some general features. But of course, when you apply it to different kinds of characters, you are gonna modify it to different extent. So, well, for example, right now I'm working to model the number of segmentation for which I do need to introduce some additional parameter and a process to accommodate this. So we still need to see how far this framework can go with moderate extent of modification.
All right, I
And
think
regarding your,
I'll have to
sorry.
cut you off there. We're running
Okay.
short on time, but thank you so much for a
Yeah.
really interesting talk.
Thank you.
All right, our next speaker is Takahiro.
Yes.
Apologies if I mispronounced your name. Do you want to share your slides?
Yes, sure. Can you see my presentation slides?
Yes, you can go ahead and get started.
Hello, everyone. My name is Takahiro. In this talk, I'd like to present our research entitled Environmental RNA Captures Community and Gene Expression Responses to Heat and Nutrient Stress in Freshwater Ecosystems. Environmental RNA, eRNA, enables analysis of community composition and gene expression under natural conditions in principle. Recent studies carried out eRNA transcriptomics in Daphne heat response and fish toxin response under controlled laboratory conditions. However, the mapping rate was low, less than 1% in both of these studies, and the feasibility of environmental transcriptomics in eukaryotes at the community level under natural conditions remains unknown. In this study, we examined the feasibility using mesocosm experiments. In our experimental design, we have two environmental stressors of interest, heat and nutrient. For temperature condition, we have ambient and heated conditions. For nutrient, we also have two conditions, moderate enriched to 40 microgram per liter phosphorus and high enriched to 280 microgram per liter phosphorus. Each of the ponds contains 1,000 liter water from Lake Hotel. There are 3 biological replicates, ponds in each combination of nutrient and temperature conditions. There are 3 and 2 technical replicates for environmental and bulk samples, respectively, in each pond. Here, the difference of the sample type reflects the difference of the size of the pore used in sampling. When preparing libraries, the technical replicates are put together. So in each pond, we have 1 environmental library and 1 bulk library. In our data analysis, we mapped 150 base pair paired end RNA sequence reads to the NCBI NR database using the SAMSA2 pipeline with adjustment. We inferred community composition using MEGAN and inferred gene expression using DSEQ2, considering the confounding effect of changes in organism abundance on identification of significantly differentially expressed genes, which we denote by DEGs, using the method by Klingenberg and Meiningen. Our analysis are mainly carried out at the class level, which we support also at the genus level. These PCA plots show the effect of heat and nutrients on the organ composition. In the top two figures, we show the result at the class level, and in the bottom two figures, we show the corresponding result at the genus level. By looking at the top figure, we find that our sample denoted by these dots are clustered by nutrient condition, which is denoted by the shape of the symbols. On the other hand, our samples were not separated by the temperature condition, which is denoted by color of the symbols. Here, the effect sizes was 0.42 in nutrient response and 0.06 in heat response in analysis of variance, indicating that the effect of nutrient on the organism composition is much stronger than the effect of heat. In the bulk sample, on the other hand, our sample were not separated by both nutrient and temperature condition. The same pattern was observed in our analysis at the genus level. Here, in environmental sample, our samples were separated by nutrient condition, but not by temperature condition. In bulk sample, we do not have separation of our sample by both temperature and nutrient condition. These figures show abundance changes in 10 representative classes, which we analyze in differential gene expression analysis. Here, the asterisk denotes statistical significance at the 5% level. The top two figures show the abundance change in response to temperature condition, where we did not find any statistical significant difference in the abundance of the classes depending on the temperature condition. In the bottom two figure, we show the effect of nutrient condition on the relative abundance of these classes in environmental and bulk samples. Here, in environmental samples, we found significant change in abundance in 4 out of the 10 analyzed classes. In particular, we note large increase in green algae chlorophyll with high nutrient condition. Here. These figures show the relationship in gene expression changes between environmental and bulk samples. Here in these plots, tested genes with adjusted p-values, and DEG denote significantly differentially expressed genes at 5% level. Here we show the result in rotifers, which were most abundant in our samples. Generally, there is positive correlation between in gene expression changes between environmental and bulk samples in all panels. And the correlation was statistically significant at 5% level in all panels. The positive correlation was especially strong when conditioned on statistical significance in both types of samples, indicating that environmental sample reliably identifies significantly differentially expressed genes in organisms. These are heat maps of significantly differentially expressed genes in rotifers. Here we show heat maps of 10 most upregulated and 10 most downregulated genes. In these heat maps, the row represents genes and the column represents samples. In these sample levels shown at the bottom of each panel, big T and little t denote heated and ambient temperature condition, and big N and D to N denote high and moderate nutrient conditions. In the top panels, we show the result in heat response under moderate nutrient condition. By looking at the analysis at the class level, we find that sample from ponds with the same temperature condition show similar gene expression pattern to each other. The same pattern was found at the analysis at the genus level, where we also found that sample from ponds under the same temperature condition showed similar gene expression pattern to each other, indicating that these genes indeed show heat response. The bottom two heat maps show the corresponding heat maps in nutrient response under ambient temperature condition. By looking at the class level analysis, we find that sample from the pond under the same nutrient condition show similar gene expression pattern, which was also found at the analysis at the genus level, indicating that these genes also show nutrient response. This Venn diagram shows the number of genes showing significant effects of heat, nutrient, and their interaction in environmental samples. Here we show result in 4 representative classes of organisms. Generally, there were much more genes showing significant effect of nutrients than heat. We also noticed that the fraction of genes showing the significant interaction effect is much higher in genes showing significant effect of nutrients than the gene showing significant effect of heat. These are KO terms enriched in significantly differentially expressed genes in rotifers in environmental samples. Here, circles and triangle denote result in Eurotatoria, which is the class of rotifers, and Brachionas, which is the most abundant genus in Eurotatoria. And in these plots, x-axis denotes the fraction of significantly differentially expressed genes among tested genes, and y-axis denotes enriched KO terms. The color of the symbol denotes the adjusted p-values, and the size of the symbol denotes the number of significantly differentially expressed genes. In the top panel, showing the enriched KO terms in heat response under moderate nutrient condition, we generally find enrichment of genes involved in metabolism, muscle regulation, translation, and DNA or protein damage repair. In the bottom plot, showing the KO terms enriched in nutrient response under ambient temperature, we generally found enrichment of genes involving energy production and metabolism. These are the conclusions in this talk. There is stronger effect of nutrients on the community composition than heat, which is captured by environmental but not by bulk RNA. This is mainly due to the large change in abundance in phytoplankton in nutrient response. There are more significantly differentially expressed genes in heat response than nutrient response. There is stronger interaction effect in heat response than in nutrient response. There is enrichment of significantly differentially expressed genes expected in heat and nutrient responses, indicating that environmental RNA reliably identifies significantly differentially expressed genes in organisms in these stress responses. Thank you for your attention.
All right, thank you so much for that interesting talk. I think we should go